All right, guys, so today I'm going to be talking about how do cells grow and divide. Primarily today we're going to be focusing on the difference between chromosomes, chromatin, and chromatids. So kind of three really close words, and we'll make sure that we get you through it and ready to go and understanding each of those. You have your notes packet, and we'll be filling that in as we go. If we get to a point where you miss something, please just pause the video and make sure you get that filled in. Sorry, you might have heard my dog whining there in the background. Had to let her outside. So the plasmid, again, produces proteins of a particular interest that a biotech company can use, and they are currently using that to produce insulin for diabetics and all kinds of neat things. And we'll talk more about those later. So, let me erase this real quick before we go on. So prokaryote or bacterial cell reproduction is really simple. So you just start with a single cell and it will grow, and then the cell duplicates its DNA. So you can kind of see in this circle right here that it's making two copies of its circular DNA. And then the cell just pinches together, and then you end up with two new daughter cells that have formed. And so the process is just that the cell goes through growth because it has to have enough cytoplasm and everything to have enough for two cells. Second step, the DNA duplicates. Third step, the cell divides. So cell division. The entire process is referred to as binary fission, and I underlined the prefix there, bi, because just like bicycle, what does that mean? Bi means two, so it just means that it's producing two different cells. So again, I'll ask you real quick, is this type of reproduction sexual or is it asexual? Oh, you guys got it right. It is asexual reproduction. All right, so your next chart that's down below, I went ahead and put the definitions on the left. On your chart, they're at the top, and you'll see where I wrote chromatin and a dash. So right next to that, you'll write DNA wrapped loosely around protein, which are called histones. And then underneath is where you're going to draw this. It doesn't work well on my slides to put it vertical, so you'll put it right underneath. So chromatin, again, go ahead and fill this in, is DNA wrapped loosely around protein. And the name and the word that they use for this particular kind of protein is called a histone. So what I'm going to have you do first is just draw these circles here. Draw 4 of them kind of together. It looks like a little group of eggs or something. And those are going to be representing the histone. And then what I'm going to do is add in the DNA to show how it wraps around it. So here's some DNA. I added some additional histones, and I'm going to go ahead and label that as DNA. All right, the last kind of vocabulary for this section, so we have chromatin, new vocabulary, we have histone, new vocabulary, and there's one more, and that is called a nucleosome. And it's kind of this little piece where it's got the DNA wrapped around the histones. That piece where it kind of forms this loop is what they call a nucleosome. All right, so you can pause this if you need a little bit more time to get your diagram or your definition done, but I'm going to go ahead and move on. So, a little bit about chromosomes, which form when chromatin starts to get really tightly wrapped around the histones. So when it's all loose, we call it chromatin, but when it all becomes tightly coiled, and this is where I'm going to go back and just show you something, nothing to write, right here. So you've got your nucleosomes, and then those become coiled, and then the coils become looped, and then the loops get even more tightly looped, and then eventually you'll see a chromosome. So that is what we would call the difference between chromatin and chromosomes. They're basically the exact same structure. One of them is loose and one of them is tightly coiled, which we see that in another place. We see that with water, right? Because if it's frozen, we call it ice. But if it's not frozen, we call liquid water just water, or if we boil it and it's in the form of steam. So it's all H2O, right? But we have 3 different names for it. That's kind of what happens here. If we have the DNA and the histones and they're loose inside, then we call it chromatin. But if it's tightly coiled and we can see it, then we call it a chromosome. Okay, so back to our definition over here. Skipping through, whoopsie, went too far. So this is what a chromosome would look like. So I'm going to go ahead and just tell you the parts to it. So when it's condensed, it's a chromosome. And so at one end, you have a telomere. And telo basically means end, right? So, or final. And then in the middle, we have a centromere. And I drew that chromosome for you. And so you just have to label the telomere, centromere, and then of course the other telomere. So just for human interest, how many chromosomes do you have? Well, you've got 46 chromosomes, and you got 23 of them from mom and 23 of them from dad. And so this right here is a picture of all the different kinds of chromosomes that an organism could have, that's actually a human organism could have. And so I'll kind of put a highlighter in here. So if this one is the one that came from dad, then this one is the one that would come from mom. So they're the exact same chromosome. Maybe it's the chromosome that has, say, blood type on it. And from dad, you got one, and from mom, you got the other, but maybe dad's blood type was A, and mom's blood type was B, and so now you have AB, and we're going to get a really cool lab where we'll do a blood typing lab when we get to genetics. And so I know there's lots of questions about, well, how does that happen? My mom has O and I have A. How does that happen? And we'll talk all about that. But it's just showing that you have 2 different versions of the exact same chromosome. So we have 23 pairs. Sometimes when they're talking about that, they would say that humans are what we call 2N, because we have 2 of all of our chromosomes, and N would be the number of chromosomes. So we could say 2N equals 46, just kind of putting it into a mathematical equation, where 2 equals the number of copies and N equals the number of different chromosomes. And we are interested in biology in all organisms, and so not all organisms are set up like that. There are some organisms, especially plants, that can be 5N, and they've got 5 different copies of chromosomes, which is part of the reason why we see so much variety in flowers, and even like roses have got all different colors and shapes and kinds, and that's because they have this great genetic variety of being 5N. So again, even fruit flies don't have 46. They have a much smaller, what we would call genome. They have a total of 8 chromosomes, but they do get, one copy from mom and one copy from dad. So for a fruit fly, we would say 2n is equal to 8. Whoop, this isn't showing up here. It's equal to 8. So then, of course, what would n be? Divide by 2, and that would be that they have 4 different chromosomes. So not quite as many as we do. All right, so I wanted to show you the chromosome when it's unreplicated, when it has not been copied, and when it has been copied. So I just took my chromosome, and to make it kind of showing this chromatin all coiled up, just make a whole bunch of squiggles, circle, circle, circle down one side, and that would be what we would call an unreplicated or uncopied. I probably should have put uncopied in front there. An uncopied chromosome. And then when the cell gets ready to divide, it needs 2 copies. So it first just makes it, but they stay attached at that middle piece or the centromere. And so now you can just make a whole bunch of squiggles, but make it, kind of connected in the middle. And when you have 2 copies that are connected in the middle at the centromere, then you would call that sister chromatids, or 2 copies connected at the centromere. And I'll highlight real quick what each sister chromatid is. So this is one sister chromatid, and this is the other sister chromatid, and they're just connected at the centromere right there. And so now the cell has 2 copies of this same chromosome, but they're connected. Same thing, just a different picture of it. All right, last definition for today. These are homologous chromosomes. So remember how in the picture where I showed you the 46, we have 1 chromosome from mom and we have 1 chromosome from dad. So in this diagram, I made the red one the mom, and I made the blue one the dad. And so I'm going to have you start way over here on the left side, and we'll make a whole bunch of squiggles to show an uncopied chromosome, or 1 chromosome that has 1 chromatid from dad. So just make a whole bunch of squiggles real quick, and that will be our single chromosome. And then it replicates, which means that it makes a copy. So the DNA makes a copy of itself. And then we end up with a chromosome that's duplicated. So both of these are chromosomes, but this one happens to be duplicated. And it consists of 2 sister chromatids that are connected at the centromere. So right here, chromosome with 2 chromatids from dad. And so these are what we would call the sister chromatids that are connected. So the whole thing is a chromosome made of 2 sister chromatids. Now we'll do the same thing with the one from mom. I'm going to change colors here real quick so you can see me kind of diagramming this as we go. So right here, we're going to make the other chromosome. This one will be from mom. It replicates. And now we have a duplicated chromosome, or a replicated chromosome, that consists of 2 sister chromatids. And I'll have you write that underneath. Here's a chromosome with 2 chromatids from mom. And again, those are called sister chromatids. Okay, so why did I have us redo these chromosomes? Because now we have a new term that means something really important about these. So remember how I had said that these are the same information but two different versions. So I had given the example of blood type.